and even the metastases, wherever they've spread in the body, continue to function like normal thyroid tissue from which they've arisen from. So they take up the radioactive iodine. And again, he couldn't image it, but he treated patients with really end-stage metastatic thyroid cancer and was able to cure some of these patients. And remarkably, this treatment is still used today, 85 years later, for both overactive thyroid and for thyroid cancer. And I think it might actually be the first example of a targeted therapy in all of oncology.
Welcome to the Oncology Insights Podcast. I'm your host, William O, and I'm really delighted to have as our guest today, Dr. Michael Hofman. I'm going to ask Dr. Hofman to introduce himself in a minute, but he's really a world leader in the use of Theranostics for cancer, and we're very excited to talk about where this field is now and where it's going, because I think it's already impacting patient care, and his center and he himself has been a real leader in developing this treatment worldwide. So welcome, Michael. Thank you for joining me.
Yeah, thank you so much, William. It's a real pleasure to be with you on this podcast. I'm a nuclear medicine physician currently in New York at Memorial Sloan Kettering Cancer Centre doing a sabbatical, but usually out at the Peter MacCallum Cancer Centre in Melbourne, Australia, where we've been doing Theranostics actually with Lutetium for almost 20 years. Since 2005, we've been using Lutetium to treat neuroendocrine tumours, and even longer for treating other conditions like thyroid cancer. And I think on today's podcast we'll be deep diving on this area of medicine that's super interesting.
Yeah, you and I were just at a celebration of the kind of founding of Theranostics. I think people will be interested to hear about that history, and particularly because it was so long ago, and yet the modern version of Theranostics is very, very exciting. So maybe you can tell us a little bit about that history and why it took so long to continue to develop drugs in this space.
Yeah, so I think many people think that nuclear medicine is a new field of medicine, but it's actually been around for a very long time. And just a few weeks ago, we celebrated the 85th anniversary of the first administration of radioactive iodine for treating thyroid conditions. And this was done when Saul Hertz, an endocrinologist who was investigating thyroid metabolism out at MGM, where there was the first medical cyclotron, he realized that the only organ in the body that used iodine was the thyroid gland. So he asked his medical physicists, could they make iodine radioactive? He thought this would be a way to treat benign thyroid conditions like overactive thyroid and also thyroid cancer. And indeed, they came back to him several months later and said, yes, we can make radioactive iodine. So this is an unlabeled product. It's just iodine, but in a radioactive form. And when it's given orally, it's absorbed through the bowel and localizes exactly to the thyroid gland. Back then, he could not actually image it. He could just use some external like gamma counters to put them over the body, and he could see that actually it was localizing to the thyroid gland. And then he gave higher doses to treat a number of patients initially with overactive thyroid conditions like Graves' disease or multinodular goiters, with good responses because they could then measure that thyroid hormone levels were coming back to normal. And subsequently, it was given to patients with metastatic thyroid cancer. And even the metastases, wherever they've spread in the body, continue to function like normal thyroid tissue from which they've arisen from. So they take up the radioactive iodine. And again, he couldn't image it, but he treated patients with really end-stage metastatic thyroid cancer and was able to cure some of these patients. And remarkably, this treatment is still used today, 85 years later, for both overactive thyroid and for thyroid cancer. And I think it might actually be the first example of a targeted therapy in all of oncology. We think about hormone suppression, anti-estrogen treatment in breast cancer, tamoxifen as an example of a targeted anti-cancer treatment. But I don't know, William, can you think of a targeted cancer treatment that predates radioactive iodine 85 years old. I'm not sure there is one.
I will say maybe prostate cancer, androgen, removal of the androgen ligand probably is close, right? Timing-wise.
It probably is close. And it's interesting how endocrinology comes back as a theme amongst these targeted therapies in cancer, in oncology. But since then, over the last 85 years, this was an example of an unlabeled radioactive substance. You're simply giving iodine in its radioactive form, not labeled to anything. In the last few decades, we've seen enormous advances in chemistry, ability to radiolabel either antibodies or more recently peptides, very small chain amino acids, to radioactive substances via chelators. The chelator is the bond between either the antibody or the peptide and the radioactive substance, so that they're tightly linked together. And this is then a way to deliver high doses of targeted radiation to sites of tumours, targeting specific receptors on the cell surface. And this has been a real paradigm change in a number of different malignancies.
Most teams don't have a strategy problem. They have a systems problem. Tasks scattered across tools, goals buried in spreadsheets, and metrics no one actually checks. Zentrix OS fixes that. It's a true business operating system that brings tasks, goals, and performance metrics into one connected workspace. So your team always knows what to do, why it matters, and how they're tracking. No more juggling apps. No more missed accountability. Just a clear, organized way to run your business the way it was meant to be run. If you're ready to stop managing chaos and start managing with clarity, Zentrix OS is built for you. Visit zentrixos.com and take control of how your team operates.
We're going to definitely dive deeper into that. But before we do, some of the characteristics of iodine and thyroid cancer or overactive thyroid made that an amazing treatment. And what's one we still use today? How many things do we still use today from 85 years ago? And Dr. Hertz was really a pioneer, days before IRBs, I would imagine, where, these patients had very few therapeutic choices, and he was able to cure some of these people, and we're still doing the therapy today. What took so long? We're going to come into the next last, let's say, 10 to 15 years when this field has really burgeoned and where you've been a real leader in this space. But what took so long? What were the characteristics of radioactive iodine that were hard to reproduce in the, let's say, 60s, 70s, 80s? Before you get to the chemistry per se, I want to really focus on the targets. Like iodine was an amazing target, and you pointed out why. Are these targets there and we just didn't know it? Are there targets like I brought up androgen receptor? That target's been around a long time. Why couldn't we radio label, testosterone? What was that gap? Why was there a gap? And then we'll get into how things have really progressed in the last decades.
Yeah, these are great questions. Look, it's worth first mentioning that although this is a treatment, it's tightly coupled to an imaging modality, either on a PET CT scanner or a SPECT CT scanner, depending what type of radiation we're giving. And 85 years ago, there was no way to really image radioactive iodine other than putting a probe outside the body over the skin and literally hearing the buzz from the gamma counter. But nowadays, and iodine is case exemplar, we can take iodine 131 or iodine 123 and put them on a gamma camera, a standard nuclear medicine camera, or we can take the positron emitting version, which is I124, and put patients in a PET camera, and we can get whole body three-dimensional images of the distribution of the radioactivity. And on the PET scanner, it's coupled to a CT, a PET CT, so we can localize exactly where that radioactive uptake is, typically in color, on the background of the CT anatomic template. And when we do this with I124, it's really an exquisite radiotracer for imaging. We can visualize metastases actually smaller than 1 millimeter. Sometimes it's just a tiny dot on the CT, and on the nuclear medicine component, it's lighting up very brightly. And we can measure this using a number called SUV, the standardized uptake value, which is kind of the ratio of the uptake in the cancer compared to the normal organ background activity. And we can also see whether there are any normal organs that are taking up the substance. Radioactive iodine is a near perfect theranostic because it has very high tumor uptake, SUVs over 1,000 at times, so more than 1,000 times background activity with almost no normal uptake, normal organ uptake. Very often when we radiolabel a substance and we give it to a person, it may have very high uptake in the stomach or any number of different organs, the liver, and this means that in addition to delivering very high doses of radiation to the tumor, we also target normal tissues. So particularly the PET CT scan has allowed us to label various substances, image it, and really predict, will this be a useful therapeutic option because it has high tumor uptake, low background uptake. If we take FDG as an example, radioactive sugar, which is our bread and butter tracer that we use in oncology for imaging any number of cancer types, this is not useful for therapy. It's great for imaging, but it's got, high uptake in a lot of normal organs. So we really don't want to irradiate those tissues, but it's great for imaging. And, sometimes we find a great imaging agent and it may not be good for therapy, but sometimes we find one that's both good for imaging and therapy. If we wind back maybe 25 to 30 years, the somatostatin receptor on the surface of neuroendocrine tumors is one that emerged as a suitable target for both imaging and therapy. Initially, it was labeled with indium-111, which is a SPECT tracer, so we can put them on a conventional nuclear medicine camera. We get somewhat blurry images with that technology, but we can still see the tumor uptake and the normal organ uptake very nicely. And more recently, due to advances in chemistry, it was labeled to gallium-68, a positron emitter. And we get beautiful images of neuroendocrine tumors with gallium-68 dotatate. And then we can change from the gallium-68, the positron emitter, which is really a very low radiation, passes out of the body. The PET camera produces these remarkable images, and radiolabel it to Lutetium-177. Lutetium-177 is a radiometal, very similar to gallium-68, and we can use very similar linker. So we can image it with the gallium and then treat with Lutetium-177. It has very similar properties to iodine-131 as a therapeutic. It has a beta particle which travels around 1 millimeter. So when we give it in a high dose intravenously, it really travels around the bloodstream, locks onto the surface of those neuroendocrine cells where it's actually internalized into the cell, builds up, deposits high energy radiation that travels only 1 millimeter. That's a very targeted form of radiation, and it's a very nice radiotracer because it has relatively low uptake in normal organs, enabling us to get, sometimes many hundreds of gray to individual tumors with almost no radiation to most normal tissues.
So it took decades, but I see the imaging component and the idea that these markers are expressed more exclusively on tumors at a high level allowed you both to image and then subsequently replace the imaging marker with a therapeutic marker, in this case Lu-177. When was the Lu-177 somatostatin treatment approved in the world, in the United States?
Yeah, look, I think it was approved. I don't have it in front of me, but it was around, I'm going to say 10 years ago, which is not that long ago. And this is an example of a treatment that was, available in Europe and Australia as a treatment for probably 15 to 20 years before it was FDA approved. It followed a bit of a alternative development pathway being developed in academic nuclear medicine departments as opposed to the, medical oncology pharmaceutical company world. And that really resulted in a little bit of a delay to widespread availability because this was a product that was made typically in hospital radiopharmacies. So Lutetium-177 is made in a nuclear reactor, and there was about 5 nuclear reactors around the world making Lutetium-177. It has a half-life of 7 days. So every 7 days, the radiation is half of it's gone. So you can ship it around the world. And then typically the lutetium would be labeled in a hospital radiopharmacy by a radiopharmacist to the somatostatin target, and then it would be given to the patient. This really limited its availability to academic centres And although it was highly effective, it had never followed a phase 1, phase 2, phase 3 trial process. So for a regulator like the FDA that wants to see randomized controlled data showing that this is more effective than a existing standard of care, that data wasn't available 20 years ago. So those trials needed to be done. And once those trials were done, which is the famous NETA-1 trial, that really resulted in a lot of industry interest. And Novartis, one of the large pharmaceutical companies, then commercialized centralized production of Lutetium-177 dotatate. And this was the beginning of, I would say, the commercial world of Theranostics, where this product could be made en masse, shipped around the world in a syringe, ready to be given to patients globally.
This is really a fascinating part of the development of radioligand therapy or radiopharmaceutical therapy. By the way, do you have a favored term for what these drugs are? Are they Theranostics? Are they RLTs? Are they RPTs, which is radiopharmaceutical therapy? Does the field have a favored terminology for it?
Yeah, look, I think there's a few different terms and it can get a bit confusing. I quite like Theranostics, although it doesn't necessarily mean radioactive targeting, but when applied in nuclear medicine, Theranostics being a word that combines the therapeutic, and the nostics being the diagnostics. So we can do both diagnostics and therapeutics, but you've alluded to the other terms, radioligand therapy, which is when we've got a ligand like PSMA or dotatate that we're radioactively labeling. The Society of Nuclear Medicine in the US has adopted the term RPT, radiopharmaceutical therapy. So a number of different terms. They really all mean the same things. But I think it would be good if we standardize our terminology, particularly for patients, so that this can be readily understood.
Yes. And, I'm a prostate doctor. I know you've done most of your work in prostate cancer as well. So we're going to switch to prostate cancer as a much more common disease, obviously, than neuroendocrine tumors, and with a real global impact. But I wanted to start with an interesting drug, which is still available, which is radium-223, which is also, I think, a theranostic or radiopharmaceutical therapy. And maybe you can discuss what's different about radium, which I think was approved now about 15 or 16 years ago, what the difference between an alpha and a beta particle might be, and why radium is different than the Lu-177 PSMA drugs that are now available and that are still being developed. Could you discuss that a little bit?
Yeah, this is really a great place to start. So radium-223, if you look at radium on the periodic table, it's in the same column as calcium. So it's a calcimomimetic. And similar to radioactive iodine, it's actually not labeled to anything. It's just radium in its radioactive form. And being a calcimomimetic, it's taken up into the bone matrix similar to calcium. And you can image it essentially with a bone scan. A bone scan is essentially showing us the biodistribution of radium. And we know that patients with prostate cancer, but also breast cancer and other cancers, have osteoblastic metastases that light up very brightly on the bone scan. And many years ago, it was postulated that radium-223 would be an effective agent for treating bone metastases. Before radium, we had strontium and samarium EDTMP, two other bone-targeted radioactive treatments, which actually had been used for many decades. When I did my training, I did a lot of treatments with strontium, samarium, and radium-223, and it worked very nicely. Patients would come with bone pain, quite severe sometimes, and you would give radium-223 or strontium, samarium, and it would be remarkable how quickly patients' bone pain would improve. Now, radium-223 is quite unique because it is an alpha emitter. To date, when we talk about radioactive iodine, radioactive lutetium, or strontium, samarium, these are beta emitters. So broadly in nuclear medicine for therapy, we have three types of radioactive emissions we can use for therapy. Beta has a fairly high energy, so we can kill cancers, but it's moderate. We can measure this using a number called the linear energy transfer, LET. That kind of measures the amount of energy deposited as the radioactive particle passes through tissue. And with lutetium-177 or radioactive iodine, it's modest. This means, similar to chemotherapy, it'll actually affect rapidly dividing cells, which are more sensitive to radiation damage, more than normal tissues, which is actually great for cancer. But it causes predominantly single-stranded DNA breaks and sometimes double-stranded DNA breaks. Tumours also have capacity to repair themselves when the DNA is damaged, and we don't want that to occur. Radium-223 is an alpha emitter. Alpha emitters actually have several orders of magnitude higher energy than a beta emitter, but they travel a very short distance. The energy is so high that it will really kill anything in its path, whether it's normal tissue or tumour tissue. So it could be very dangerous. And before radium-223 was shown to be effective, people were actually very scared of using alpha emitters in humans. But a famous trial, the Alsimca trial in prostate cancer, that randomized men to either radium-223 or standard of care in patients with very advanced cancer, showed that radium-223 was not only improved survival, but was also very, very safe. And when we look at its biodistribution, quite similar to radioactive iodine, it's got very high uptake in these osteoblastic metastases and a very low uptake in normal organs. You see this when you do a bone scan. You see high uptake in the tumours, relatively low uptake everywhere else. However, it's important to bear in mind that this is targeting the interface of the tumour with the bone, the osteoblastic interface. It's not actually a tumour-targeted therapy directly. And of course, prostate cancer can spread to other sites beyond bone in lymph nodes, other soft tissue sites, liver, lung, etc. And radium-223 will only target the bone metastases. So it does have limitations, but nevertheless prove that alpha therapy is a safe therapy in humans and a treatment that's still used today and still of widespread interest.
Yeah. And this is not the same radium that Marie Curie used or discovered, is it?
Correct. It's a different isomer of radium. So this is radium-223 in its alpha form.
Okay, much shorter half-life.
Correct.
Yeah, not thousands of years. So let's move ahead to Lu-177 PSMA, which you had a major role in developing. And, you brought this up with regard to dotatate, which is that it was really led by academic centers, which is really fascinating because it's just such a very different development approach than what we see. Like, I'm at Yale. I wouldn't think I could develop a drug here at Yale. I don't have manufacturing. It's not the kind of thing that any academic site thinks it can do. But yet for at least a decade, I was sending patients to Germany because it was the closest place, doing these in-house radiopharmaceutical programs because they were publishing very small series. And you were really the first one to do across, both in your institution at the Peter Mac, but also across Australia, these much larger proof of concept clinical trials, really funded not by industry, but funded by philanthropy and by your institutions themselves. So first of all, talk about Lu-177 PSMA, what that development pathway was and where that particular drug is now.
Yeah, so PSMA is prostate-specific membrane antigen. It's a cell surface receptor that sits on prostate cancer cells. It was actually cloned right here in New York out at Weill Cornell and Memorial Sloan Kettering well over 20 years ago, and antibodies were made against this target, and efforts were made with radiolabeled antibodies, both for imaging and therapy. And they had modest utility, either as an imaging agent or as a therapy. The very first versions of the antibody actually targeted the intracellular domain, which was not very available. Then some newer antibodies targeting the extracellular domain were developed. But radiolabeled antibodies is a little bit tricky. It takes some time for the radioactive molecule to bind to the cancer after administration. They have a very long circulating half-time. In a collaboration, really, again, between Weill Cornell and University of Heidelberg in Germany and others, some groups developed some radiolabeled peptides. so small chain amino acids, rather than an antibody that binds to the extracellular domain of this PSMA receptor, similar to dotatate in neuroendocrine tumors. This is a target that I first discovered, I think, in 2011 at the European Association of Nuclear Medicine meeting, where a lot of great nuclear medicine science is presented. And to me, it seemed to have the characteristics of an ideal Theranostics, so very high tumor uptake and very low background uptake. So I remember going back in those days, we were interested in fluorocholine for imaging prostate cancer, and also just fluoride as a PET bone agent. And this PSMA was coming along, and it looked very exciting, both as an imaging agent labeled to gallium-68 or as a therapeutic labeled to lutetium-177. And we had a very large program in our centre for using gallium and lutetium in neuroendocrine tumours. So it was very easy for us to take this platform. We had the in-house radiopharmacy, the infrastructure to do it, and label it to PSMA. And I decided, rather than doing it as a compassionate access therapy, which is what was happening in Europe in men who had really failed other treatment options, that we would, do more medical oncology style clinical trials to prove that it works, both as an imaging agent and as a therapeutic. Because what I learnt from my neuroendocrine experience days is that the oncology world often did not believe the relatively poor quality data that we produce in nuclear medicine. We would produce beautiful images showing that this new imaging agent localizes a particular tumor, but the medical oncologists and health regulators would come back and say, well, these images are great, but how much better is it than a CT or a bone scan? Or as a treatment, how much better is it to an existing standard of care? So the very first trial we did was actually the Pro-PSMA trial, a imaging trial funded by Movember and the Prostate Cancer Foundation of Australia, an academic trial that we ran at 10 sites around Australia. And we took men with newly diagnosed prostate cancer, intermediate or high risk, and they were randomized to either a gallium PSMA PET scan or a CT plus a bone scan, 2 scans. And in 300 men at more than 10 sites, we were able to show vastly improved accuracy, a high management impact, lower radiation doses. This was published in The Lancet in 2020. I think the manuscript now has over 2,000 citations and really set a benchmark for proving that this was better than either a CT or a bone scan. Around the same time, we also ran a very small 30-patient trial of lutetium PSMA 617. This clinical trial treated its first patient in 2005, 2015, 2015. And back then there was not much industry interest. We were lucky enough to get lutetium supply from a reactor in Australia that produces radioactive lutetium. We were able to get PSMA 617 from a company in Germany called ABX, which had licensed the molecule from University of Heidelberg. We labelled it in our hospital radiopharmacy, and we gave it really under a strict clinical trial protocol in collaboration with our medical oncologists, urologists, and radiation oncologists to 30 men who had no other treatment options available. And we were able to show remarkable responses. So more than a 50% drop in PSA occurred in 64% of men, which is really remarkable in this end-line setting. And we were able to also show that it had relatively low adverse effects and improved quality of life. We also produced some stunning images of the gallium PSMA PETs before and after therapy. And many of these patients had really very bad symptoms. They had extremely widespread bone metastases, very poor quality of life on high-dose opiates. Some of them were more like ECOG-3, really unable to perform their activities of daily living. And after a dose or two of this new lutetium PSMA 617, they were able to wind back their opiates, often stopping it completely, and just dramatic improvements in quality of life.
Every person on your team is wired differently. Some thrive with structure. Others drive results through autonomy. Some process information fast and move on. Others need depth before they decide. And when the wrong person is in the wrong seat, not because of skill, but because of how someone is built, performance suffers and no one knows why. Zentrix Insights maps exactly that. Behavioral patterns, cognitive style, and natural work preferences are all visible in 15 minutes. So you stop guessing who should own what and start putting the right people in the right seats. Because the best teams are not just talented, they are well placed. Visit ZentrixInsights.com.
It was amazing. And I want to fast forward because I want to spend the last couple of minutes talking about the future. But of course, ultimately, pharma did recognize this on the basis of your early studies, which were quite amazing when I saw some of that data. And of course, when we started treating patients ourselves. But of course, there were two randomized large clinical trials that led to the approval of 617 PSMA, LU177 617 PSMA therapy in first post-chemotherapy and then pre-chemotherapy, what we call castration-resistant prostate cancer. And it's been widely available now for the past few years around the world. And in fact, what's amazing when you tell this story is how recently these studies were done. I mean, this is a really, really active area. And I know for a fact that I have helped many patients now with this treatment, both off protocol, and of course, we're trying to improve it with combinations and newer versions of this. So really, it must be incredibly satisfying to be so important in the development of this drug and this class of drugs, I should say. So I want to spend the last couple of minutes, really, Michael, asking you to talk about the future. This is an amazing development in a relatively short period of time, going back to when you saw the imaging and you saw the value of 617 PSMA as a way to see the cancer. And then, like the iodine example, you said, if we link this to the right type of agent, we can actually improve survival, which is what has been turned out to be true. What is the future, both in prostate cancer, but much broader, about this class of agents in your mind?
I think, as you've alluded to, this started as a last line of therapy. Now it's moving back to a earlier line of therapy. And, I suspect in selected patients in the future, hopefully we'll have some data that it could be a first-line therapy. It may even be able to delay hormone treatment in some patients. Maybe if we combine it with surgery or external beam radiation, maybe we can even improve the cure in selected patients, because with radioactive iodine, we can actually cure patients with metastatic thyroid cancer. So that treatment's going to move earlier and earlier and combined with other treatments. But it has really opened Pandora's box. What other cancer types can we treat? How else can we apply this technology? And in the last 10 years, we now have more than 50 medium-sized biotech companies involved in this space, many billions of dollars now invested, and various approaches being taken with different targets, peptides, antibodies, nanobodies, and different radioactive substances to try to apply this technology beyond thyroid, beyond neuroendocrine, beyond prostate cancer. And it is not easy to find these molecules that have super high tumor uptake and very low background uptake, but we do have the luxury of being able to image it on a PET scanner and predict upfront whether this is likely to be effective. So we can take a shortcut rather than just randomly trying it. We know that most phase 1 trials in oncology fail. Very few make it through to phase 2, phase 3, and regulatory approval. But here, with our imaging, I think we actually have the capability to screen and have a stop-go. I mean, if we're not seeing super high tumor uptake and low background uptake, I say, well, let's pause. Let's not take this one through to a therapeutic. Let's try something else. So many targets, many radioactive molecules being evaluated in early phase trials in the clinic. And I think we're going to see a whole range of different theranostics and different tumor types in cancer, and maybe even beyond cancer in some non-malignant conditions. This approach can be combined as well. So a super interesting time for this area of medicine.
Yeah, thank you, Michael. It's just really exciting. And like with all new areas of drug development, which is really an old area of drug development that's getting its second wind here, There's always barriers, and we don't have the time to dig into all of those risks and barriers, but thank you for all the work you've done in this space, for being an international leader, and we're really excited about where this field is going to go and your role in it. Thank you for your time and for explaining this to our audience.
My absolute pleasure. It's a real pleasure to join your podcast.